Well, hello, and welcome back to Bio 212 Final Exam Review, FER. That's how you'll find it on the YouTube channel, if you've already found it. This is going to be the second part. It'll be the remaining slides of how I'd like you to be thinking about preparing for the final exam. And so far what we've done is more along the lines of taking a big step backwards, looking at how the human body is built. Notice I'm not drawing a lot right now, but I'm going to try, I'll switch gears to sort of put the types of information together. So we can take a look at some of the other sections in the review sheet. But notice I'm not going through them in the specific order. I don't want you to read too much into that. I would think that right now, though, that breathing is a pretty good thing that's required to stay alive. And if you don't do it, not only will you die, but at this point in time in your career inside this class, you should be thinking, why would you die? Right? If you can't breathe, carbon dioxide levels get to be too high. The pH inside the body drops. There's a specific word for that, right? Why is it called acidosis? And if it's respiratory acidosis, why? What caused it? Okay? So, moving on, you're going to notice that if we go from respiration, we're now going to take a look at what happens when we actually have to push fluids around the body. Okay? And you know fine well that I want you to be thinking, first of all, let's change colors here so I can use the appropriate colors later. Okay. Matching categories that are going to increase or decrease heart function, right? So I'm going to go like this, I'm going to go like this, and just thinking about it quickly, make sure you address all of the bullet points in any one of these sections, right? So the first part is going to be matching categories with choices that increase or decrease heart rate. Well, what increases and decreases heart rate in humans? There's more than one thing. But notice right down here, look at this. Okay. Describe the stimulation of the heart to sympathetic, parasympathetic, vagus, or cardiac accelerator nerves, as well as the neurotransmitters that they use. Remember, I've put this on the board for you before. Think about what will, the way I've drawn it for you in the past, what increases or decreases heart rate. Okay? Because increasing and decreasing heart rate changes the flow of blood to the lungs, return of blood to the left atria to the left ventricle then out to the systemic circulatory system, right? Now, notice I did that all in. The indicating color here was black. When I switch gears, you need to be able to understand that the right side of the heart is deoxygenated blood, the left side of the heart is oxygenated blood, and the reoxygenation occurs in the capillary beds of the lungs. And remember, this is a portal system. So, sequence the blood through the valves. We just did it. You didn't even notice it, right? So this is the right atria to left atria. What is right atria. That was so wrong. I'm so sorry. Right atria to right ventricle, right? What's the vessel? What's the literal valve that it's passing through? As we go from right ventricle to the lungs, the pulmonary trunk, what valve did we pass through? You know, this needs to be second nature at this point in time. I'm going to get rid of some of the cartoon right now. I'm going to draw another heart. So let's go like that. Get rid of that. Let's start all over again. Let's draw a different heart this time. Okay, and here's what I want to do. I just want to go like this, sort of like I did in class, multiple times. Here's our square. Here, now let's change some colors. Let's go like this. So you can see the conduction system. I don't know if this is going to, that doesn't show up so well. Let's change colors. Let's go green. I'm going to go sort of pink red. There we go. That's going to show up. Very good. Fantastic. SA node. AV node. Bundle of His. Notice it's inside the actual intermuscular layer that is cardiac myocytes, right? We have our Purkinje fibers, and there is our entire conduction system. Okay, SA goes to AV, AV goes to bundle of His goes to Purkinje fibers, and voila. Now remember, this is regulating the muscular contraction. So it only makes sense that we ask you, what area of the brain regulates the heart. And I'm hoping immediately you come up with medulla, right? Med. Okay, cool. So if this is the heart, and we know we needed to pump fluids throughout the body, there's a consequence to knowing that if we change heart rates, let's go cardiac output. Oh, yeah, there you go. Cardiac output is heart rate times stroke volume, right? It's a V. V. Okay. Doesn't look like a V. I apologize. Erasing is a pain in the ass, so this happens. I'm going to erase the whole thing. Okay, but you get the idea. If we change anything here, it changes what happens inside the capillary bed. Okay, Mother Nature has the ability to regulate what happens there. But what I want you to do now with your heart here, right? Hey, look, we can go back here. Sympathetic effect on heart rate. Parasympathetic effect on heart rate. Vagus effect on heart rate. Okay? And if your brain is going, well, they're the same goddamn thing, well, then you know that, right? Once you learn it for one, you've learned it for the other. Here's where some people will make a mistake. Acetylcholine and norepinephrine. Remember, acetylcholine is being released by the vagus nerve. Norepinephrine is being released by the cardiac accelerator nerve. Okay? With me? Cool. So, let's get rid of these cartoons and move on to the next one. Oh yeah, and this. So, you're going to have a matching category that has an artery, a vein, a capillary, an arterial, a venule, and all of the above, and none of the above. And where would we see all of these? Or what are they doing? What are they? They're all part of a conduit system of one sort or another. And in this case, we're transporting blood, right? But what about, say, lymphatic fluid? What do we transport that in? Well, it's a capillary, right? It's turned into a trunk that returns fluid back into, the venous blood return. These are all conduit systems. One is closed, the other is open, is the point that I'm making, right? Can you differentiate between the types of blood vessels in terms of anatomy, function, location, and capillary beds? Okay, but if we're asking you about blood vessels, make sure you understand the construction of an artery, a vein, a capillary, an arterial, right? A venule. Do blood vessels receive parasympathetic innervation? Generally speaking, the answer to this would be no, right? Generally speaking. Which receive the most autonomic innervation? Well, those would be arteries, right? Arteries and arterioles. If you think about, say, what we see inside the kidney, at least, right? That afferent arteriole has direct sympathetic innervation. Now, remember a moment ago, let's draw this again. Okay, we're going to go back here, we're going to go here, we're going to go back to this, and look, here is what I was talking about a moment ago. If cardiac output is this, HR, we do anything to, just the dot multiplied by, okay, stroke volume. We do anything that changes stroke volume or heart rate, then we're going to change cardiac output. But notice what's actually happening now. If the consequence is to increase these, what effect does it have? Whoops, not underlining. It's supposed to increase blood pressure or decrease blood pressure. Okay? So anything that reduces heart rate should decrease blood pressure. Anything that decreases stroke volume should decrease blood pressure. If I threw a curveball at you and said, oh, if I apply atrial natriuretic peptide to the human body, what should happen to blood pressure? Well, it should drop, right? What do we mean if we apply the stimuli from below? Okay, so notice we are speaking specifically about sympathetic, parasympathetic, acetylcholine, and norepinephrine. And again, I'm going to review this with you. Acetylcholine from parasympathetic nervous system, norepinephrine to the cardiac accelerator nerve. And remember, inside chapter 21, I made a big deal out of this. Hopefully you read it and understood it as well. Inside chapter 21, the major difference was that cardiac accelerator nerve also targets all of the arterial blood flow inside the body, whereas the parasympathetic nervous system does not. Okay, cool. Finally this, and again, These 10 slides are to help you sort of think about preparing for the final exam. You've got, I don't know, 3 days, and over those 3 days, there's probably 10 useful hours. And in those 10 useful hours, sort of break it up into 25-minute pieces of time, give a break between each one, step back, think about how you've seen this information. And that's why I said, finally this. Have you noticed we only discuss these after everything else? Okay? The reproductive systems we discuss, even though they're the first exam, and maybe this is a mistake by us, right? They should only be discussed in terms of we've moved from a child to a young adult. What's happened there? We've had a series of hormonal changes inside the body that have been instigated to change the significant tissue responsibilities inside the body. And notice here I put both 28 and 29 here. There are 11 questions probably in total, right, between all of these things on the final exam. That doesn't mean don't study it. What it means is you have to reframe how you apply the information. And by that I mean sequence of events of ovarian and uterine cycles beginning with menstruation. Well, that means that we're talking about the uterus. We're talking about the uterus after follicle-stimulating hormone and luteinizing hormone have been made. So I'm going to put those inside there. FSH and LH. And you notice those have to be made first before we make estrogen and progesterone, E and Pro. And then once you have E and Pro, then you actually drive all of the other responses, right? So at what stage... So let me just help. Actually, no, you think about what this means. It's all about timing. And the timing here is over 28 days. What's actually happening at the ovary? Increasing concentrations of follicle-stimulating hormone, increasing concentrations of luteinizing hormone. Luteinizing hormone triggers ovulation. The stratum functionalis is what's shed at menstruation. After there's no more progesterone, right? Everything becomes anoxic or hypoxic, right? What causes this? Hypoxia causes this. Hypoxia. Hypo, no oxygen. Hypoxia. Okay. Next is sequence of path of sperm out of the body. And I know we've already discussed this in lab. It was on your practical. And it is not so much that you think about, because this is the same question right here, right? This is trigger of ovulation. How do you get the secondary oocyte in a proximity where it can become fertilized? Sperm have to be made as a result of spermatogenesis, right? And then they actually have to find their way out of the body. And that means you have to understand the ductal structures, right? Because the male has a external reproductive system, whereas the female has an internal reproductive system, which means Mother Nature had to engineer a path. That's what's important. Okay, so again, this is going to be a hormone question. What hormones prepare the endometrium for implantation? Well, you can't do this without thinking about the role of estrogen and progesterone. Primarily, progesterone drives vascularization in here, right? But are there other hormones? Go back to your list and make sure you check. Once fertilization has occurred, that's the assumption when we move into chapter 29. What happens, or how do we make an amnion? Okay, what is it? Well, it is essentially waste materials, right? What is a chorion? Well, these are blood vessels that increase the surface area to collect more oxygen and remove more waste from the developing fetus. Allantois becomes, it's the progenitor structure, and becomes the umbilicus. Umbilicus. Right? The yolk sac is where we actually generate our primordial sex cells and red blood cells. Remember, these are fetal F RBCs, BCs, and remember they bind oxygen. And it's all part of, guess what? A portal system. Okay, the placenta itself is the major surface area structure that allows for gas diffusion to occur, right? As well as nutrients and waste removal, right? The last part to this, right, is the types of membranes, their anatomy, and then in what order do they grow, okay? Well, the membranes, obviously, we want you to think about. Review these first, before you review anything else. And if I'm you, you're going back either into your textbook or your lab handouts, right? I mean, unwittingly, maybe unwittingly to you, but not to me, you've created your review packet for the final exam by making those lab packets. So pull them out, look for these pieces of information inside of them. And if it's not finished, now's the time to finish it, right? With me? Okay. The last piece here, right? This is the last slide. I might throw some other thoughts in here at the end, but when we're thinking about the sequence of developmental stages, obviously there's a correct order here. Meaning it's not listed in the correct order here, but you should rearrange it so that you can do that. No different than we did on our practicals. Remember, I would take the model pieces, I would rearrange them for you and say, hey, put them back into the correct order. That's part of the reasoning behind putting lab questions together like that. So you can see that you actually have to have a fertilized gamete, right? Spermatocyte meets secondary oocyte, which gives us a zygote. And the zygote then becomes a morula. And the morula, because it's trapped inside that glycoprotein structure, gives us a blastocyst because the fluid gets trapped in there. Ultimately, we end up making a bilaminar disc because these are the epiblasts and hypoblasts to give us finally a trilaminar disc. When you say the word trilaminar disc, you should be thinking of endoderm, mesoderm, and ectoderm, right? Okay, so probably this isn't what you were expecting in terms of a review. I mean, you were maybe hoping I was going to go through each section and go, here's what you need to know here and here. You're smart students. You know how to collect this information. What I want you to be able to do is sort of apply it on an exam. So if we ask you, say, about blood cells, you notice I didn't talk about them yet, but you know fine well from this conversation, red blood cells are probably the most important cells for you to understand, right? We don't get oxygen to our individual cells. They die, pH changes, you die. That's all there is to it. Okay? That's the case. We need to get oxygen there. It means you need to understand cellular respiration. All right? We'll talk about cellular respiration in class, and we did for the last exam, but there are glycolytic reactions, there are Krebs cycle reactions with the electron transport chain, and there are transition changes, right? And what I want you to take away from this is that if oxygen gets to any one of the cells inside of here with some glucose inside of it, it's going to do what? It's going to go through glycolysis. It will then go through the Krebs cycle. It will then go through the electron transport chain to make more ATP and drive all of this. We need to get rid of wastes, okay? And the nephron is the major structure that gets rid of that. So we've gone through that a couple of times now. Electrolytes and acid-base balance we're going to come back to. But what I mean by that specifically is we need the nephron to maintain acid-base balance along with respiration so that you have the appropriate electrolytes inside of the body. And those electrolytes obviously are sodium, potassium, calcium, chloride, and phosphate. Okay. Just some closing thoughts here on how we actually think about true and false questions when we're writing them here. Okay. I want you to understand that a lot of the mistakes that I see when people are reading true and false questions, right? I'm getting updates. I'm not sure I want this right now. Let's get rid of you, even though I think you're probably going to be important, but not right now. Let's hit pause. Okay, sorry about the technical difficulty there. Just this last thought. This last thought is true and false questions. Every time I read people's true and false questions, they tend to have the same sort of mistake. And that is, you bring additional information into it that isn't relevant to the question. Start with a really basic reading of it, okay? So that you can see that if there's anything false in the statement, the entire statement is false. And that's all there is to it. At least that's how I approach them. And yeah, you're always going to think that we're trying to trick you. I don't think we're trying to trick you. That's not the way I would approach it. I would think that we're trying to make sure that you know what you're talking about. And if you don't, well then, hopefully when you leave the room, you're like, okay, I need to go back and, think about some of this stuff a little bit more rigorously. But it's been fun. I hope this has been helpful, and I will see you all later. Have a great day now.